This is the 6th lecture in Unit 1. We have some additional cell structures which include the centrosome, which will make centrioles. These are used to help pull the cell apart during cell division. We also have cilia on some cells, which are going to be multiple little hair-like projections used for cell movement. These are going to cause a crawling movement, much like a spider. Flagella, on the other hand, are going to be more of a whip-like tail that you would see on sperm and cause a swimming motion. We're going to follow the link to watch these in action. The ribosomes and endoplasmic reticulum play an important role in protein synthesis in the cell. The ribosome is the organelle associated with protein synthesis. The endoplasmic reticulum is a membrane network within the cell that comes in two variations. The rough ER, or endoplasmic reticulum, has ribosomes attached to it. These are used for making secretory proteins in membranes. The smooth ER does not have ribosomes attached to it. It functions more in making fatty acids and steroid secretions. The Golgi apparatus has a pancake stack type of structure to it. It is used for packaging and modification of the secretions produced in the cell. The individual layers or pancake layers of the Golgi are called cisternae. Some additional important structures in the cell are going to include lysosomes. These are going to be membrane-bound packages that contain enzymes to lyse cells. Most of the time this is done to protect the cell, but in some cases lysosomes will be released on the cell itself. We call this process autolysis. While it may seem like an illogical process at first, the cell is looking out for the greater good of the whole organism in most cases, and if it means sacrificing the cell for the organism to benefit, it will. Peroxisomes are another type of vesicle that will contain hydrogen peroxide. Mitochondria are yet another organelle. These are an important part of our energy production and are sometimes referred to as the powerhouse of the cell. Try to think about what your cell would use peroxisomes for. It's actually not all that different from what you would use hydrogen peroxide for. It will use it as a means of trying to kill invaders or other bacteria. Our genetic information can take on several different forms. Genes are pieces of the DNA or a region of the DNA that codes for a particular protein. Chromosomes, on the other hand, are the organized form of the genetic material. These are seen only during cell division when the genetic material is coiled up tightly and getting ready to divide into two separate components. The genome is a more generic term for the genetic information in the cell. It doesn't specify what form it is in. Chromatids are the individual legs of the chromosome. These are what pull apart in cell division and are sometimes referred to as sister chromatids. The chromatin, on the other hand, is the loosely coiled DNA. This is how the cell will spend most of its lifetime, with the DNA in the chromatin form. This picture illustrates the different forms of the DNA. You can see where the chromatin becomes tightly pulled together, it will form a chromosome. And if we look closely at the chromosome, you will see a region of the genetic information, or the base pairs, as part of that coil. This particular part of the coil that codes for the protein is the gene. The nucleus is surrounded by the nuclear envelope or a nuclear membrane, which is very similar to the plasma membrane. Inside the nucleus we have a dark area called the nucleoli or nucleolus. This is responsible for assembling ribosomes.